Hello, welcome to our lecture number 8 on single-cell high-throughput data science. Today, we are going to talk about pseudotime and trajectory analysis. And I will explain what they are and how this analysis can be done computationally based on the single-cell RNA-seq data. So let's go started from introducing the cell lineage tracing technology. So in embryonic development study, in homeostatic tissue turnover study, and also for study of stem cell and their implications in regeneration and disease study, we want to follow individual cells and see what's their development fit. We want to tracing this dynamic process and understand their development trajectory and then what their progeny are. So in modern lineage tracing technology can be divided into prospective and retrospective, two large groups, and we will see next slide about this separation of the methodologies. So for example, if you use image-based, you make a cell visible and by introducing marker or dyes, so you can follow the further development of the cells. And this is one of the prospective methodologies. You can also use genetic marker and fluorescent other coloring system as reporters to follow the trajectory of the development. So this large group of technology are prospective. Okay, so this picture is downloaded from this review paper, and to illustrate different methodologies. And if you look at the perspective from this image-based, genetic marker-based, and other marker systems, they all prospective approach that you set up your experiment and you're ready to follow the development of the cells before they start to accumulate mutation or start to differentiate. On the other hand, the retrospective is based on the detection of nature DNA markers in the population or in the pedigree system. So you sequence individuals and identify those mutations and follow the mutation, and you can somehow reconstruct the history of the cell development or evolution. So this is a retrospective method. And then in the prospective method, you can have an image-based, you can genetic marker-based, and other systems like gene editing based. So this is a overview of the different strategies to reconstruct cell lineage trees. So that's the purpose of this tracing system. Go back to motivation, why we are interested in the cell development. So this is cellular process, the cell differentiation and the maturation is a dynamic system that nature and not always will describe. You want to understand this to fully understand the regulatory control for the fate of the cells. And that's a study from for the cellular dynamics, and very important to do this in order to understand the cell development and characterize the transition between different cell stages. And we borrow this so-called robust genetics robustness, the landscape of this from Washington. And people reuse this picture, but explain it using the cell lineage tracing strategies. For example, if you have an undifferentiated stem cell, there are different trajectory you can go to develop them into differentiate them into B cell, macrophage, or fibroblasts. The fate of this trajectory is only determined by a couple of the key transcription factors. Okay, you can even reverse this process. But the picture of this show you there are several fixed canoe that you can roll down this ball from the early stage of development and into this canoes, several fixed canoes. So it's not a completely random process. It's stochastic, but still you have this underlying trajectories you have to, the cell has to follow. So we try to understand these trajectories, whether there are a lot of different ones or there's only a couple major ones. So that's the purpose of this. So the methodology we are going to use is called trajectory inference or trajectory analysis to infer this so-called pseudo-temporal ordering system. So pseudo is it's not real, the temporal is time related. So you want to build a pseudo-time trajectory. So this is a computational methodology, and using single-cell RNA-seq data, you can achieve this. The goal is to characterize the difference between cells by placing cell along a continuous path. So this path is trajectory. This path represents the evolution of differentiation of the process rather than dividing cells into discrete clusters. So you want to build a continuous trajectory instead of the clustering analysis to put cells into groups. Okay, that's a difference. They are related, but the difference, the trajectory analysis try to find the continuous paths. So that's this path, and it's where the cells are going to be trajectory projected onto. So this axis is called pseudo-time, and to represent the progress through the process. Okay, so that's a definition of pseudo-time. So we are going to use real examples to show exactly how this can be constructed and how this inference process can be done. Again, the goal of this trajectory analysis is to understand the gene dynamics later and to model the transition between cell identities of different clusters. Oftentimes, it's a model the differentiation process, identify gene associated with this pseudo-time. So that after you construct this trajectory, you want to identify genes that change their gene expression level associated along with this trajectory, and those genes associated with the pseudo-time. To infer this, oftentimes they may not be a real biological process. The reason is in a single cell is destructive process. Single cell RNA-seq has already break those there's no information about the real biological time of development. There's only a single sample, and also there's no spatial information to allow you to reconstruct. So that's why the trajectory you constructed is follow a so-called pseudo-time instead of a real time. But once you, because single-cell RNA-seq give you enough cells, you have a large sample size of the cells, so you can reconstruct this transition states very accurately because there are a lot of cells which distributed between different stage and merged from one stage to another. So you merge the cell clusters into continuous trees, paths. I have shown you this before, so I try to use this picture to illustrate the principle of pseudotime in the construction of the trajectory. For example, you want to study the movement of the horse in the gallops of this running. However, you don't have a video recorder to take a series of pictures from one horse. What you can do is to get a picture of a lot of horses, running horses, and then align them and according to the position of their similarity, and put them together, linearize them into a trajectory, and you probably can reconstruct a movement for this individual horse movement. In this way, you study this this movement by using only a picture of a group of running horses. Similarly, in our single-cell RNA-seq trajectory analysis, you obtained one sample that has a lot of cells in there. Then you rearrange the cells by along this path and assume they are separated by a transition process from one end to another. And there are a lot of cells in the middle of this transition, and then you reconstruct the development stage status for reflect a single cell process. So that's the idea. To further illustrate it, then in real development stage, you may have cells in different physical time points, but you collect this sample and pull them, and that's for single-cell RNA-seq experiment. And then in this process, you capture process, you lost this temporal information. But after your sequencing and get the gene expression information from those individual cells, and then you can use the pseudotime estimate to reconstruct this pseudotime axis, and hopefully this reflects the real physical time. And because you order the cells by the pseudotime, you can follow individual genes expression. For example, you look at the gene A's expression across all the cells, and this gene A expression level decrease along with the increase of the pseudotime, and on the other hand, gene B have increased level of gene expression along the pseudotime axis. So we try to recover those genes associated with expression associated with the pseudotime. So those genes, maybe some of these genes are driver genes, and so they drive the cell from one state to another, or differentiate from one cell type to another neighbor cell type. So this is a study in the early stage using pseudotime analysis to study the human embryonic stem cell derived into endothelium cell. So they use this technology to study differentiation. So their sample have different combinations from a stem cell to progeny and to the more differentiated stage of cells, and put them together and sequence using 10x genomics, and then clustering and all reconstruct these trajectories, and then to study which gene may associate and regulate this three different processes, because they reconstruct three trajectories, 1, 2, 3, okay, to the different directions. That's still from the same paper, and look like they identified two separated trajectory, and then there are different factors change, and then at the end they are converged into the same cell type. They identify the two different parts, from a stem cell to this more developed stage of cells. Though that's what they can reconstruct the profile to discover genes have different patterns, and to plot those heat maps to find the change of the gene expression and follow the two different parses. This is another separated study, and study using pseudotime analysis for small intestinal epithelium and differentiation. And what they used is different dimension reduction technology, and they use different technology to do this, to reconstruct the trajectory. We will mention how this can be done using real example. So now I'm going to show you some real example and to illustrate what kind of problem we will have. So this is a real data example. And for the data, if you each point is a cell, so this is a tSNE plot. And using this two-dimensional tSNE plot, you can see there's a clear trajectory along the cell population, and the cell at this end is quite different from the cell at this end, and the change from this position to this follow a trajectory along this majority of cells. Okay, so this picture is two-dimensional, but if you use higher dimensional, three-dimensional, then you will have a different view. But in reality, this is a relatively simple data set. You see, it's a clear trajectory from this end to this end, even in the three-dimensional space. So that's a relatively easy case. For instance, we can use approach. So this is a relatively simple case. For instance, we can use this to use analysis, which we can find the trajectory among the cells. So if you look at this, there is red curve. So it's implemented by a method called the spline, and this is spline fit curve, which follow the majority of this cell population. Once we reconstruct this pseudotime, we can assign each individual cells by their the time. For example, if you think this will be time equals 0, and the next step here may be times 1, and then here will be 2. Along the way, you can assign the neighbor cells to the trajectory and give every cell a time point. So this time point can be up 3. So as long as you follow the incremental steps of this trajectory, you can assign either 1, 2, 3, or whatever steps you want. After that, we can have a pseudo-time assigned for all the cells. So each point here is a cell. And then we can look at the expression of particular gene among these cells. So if you look at this green curve, the green curve is a regression in the middle of this green cells. So green cells is all the cells and arranged by their pseudo-time, and then also by the expression level of the green gene, which is this IGLC2. And you see, we can see from this step to here, from time 0 to time over 250, 300 something, and then the expression level of the green gene is decreased. On the other hand, this dark blue gene can increase in their its gene expression among cells. So this is a very simple case. You can have a clear interpretation about the cell and the development between cross stage. Let's look at a more complicated example. So this is not a so simple case. In a two-dimensional space, you see, this is not a simple linear. And if you think this will be time 0, and then you can draw a trajectory somehow, follow this spin curve. And then you may also want to have this separate trajectory. If you want to study this process more accurately, there are another 2, maybe 3 trajectories you can study. So this is more complex. Number 1, this is a tree structure instead of a single parse. So this T1 to Tn will not be the this is the primary parse, but then you still want to consider the secondary and the third parse in another place here, like here, and here, and here. Okay, when you look at the 3-dimensional of an interpretation, 3-dimensional distribution of cells, and then you will see this structure is more complex than you thought. So you may want to have a technology algorithm to follow a trajectory like this shape. Okay, I can tell you this is not so easy, and the algorithm need to be carefully tuned in order to capture this global signature about the shape of the shape signature. So we are going to go into a little bit of details and see how this trajectory can be found, can be constructed. So that's the key for trajectory analysis. Okay, so when we visualize the population of cell, we use dimensionality reduction technology. So that's the key steps, and that's also this algorithm for trajectory analysis. That's the foundation. So a lot of those Pseudotime or trajectory analysis software packages are based on dimensionality reduction. And the first step is to use one of the dimension reduction algorithm like ICA, TCA, or tSNE to visualize cell populations. Then those algorithms use different ways to find the parts. So one is called MST. Okay, and then you can also use curve fitting. So we are going to, next I'm going to mention what is MST and what kind of curve fitting and we can use. Based on the different options, you create different parts, okay, create major parts, and then rearrange, move your cell, project your cell onto these parts, and then finish your trajectory construction. So let's see what's the step of curve fitting and MS. MST is called minimum spanning tree. So minimum spanning tree was called, it's a subset of the edges of a connected. The edges can be weighted in an undirected graph that connect all the vertices together. And so vertices is those nodes. So in this example, you have 10 nodes, so 10 vertices. And their close neighbors are linked. So like this and linked with this and have a weight 6 and linked with 4. So the algorithm of the MST, minimum spanning tree, is try to find this tree structure to link all vertices together, but using the smallest distance among those linkers, the smallest sum distance. So that's the algorithm. So once you're finished, you have this tree structure which can allow you to go from any of these vertices to any other vertices. So there's a completely connected tree. Extract out of this network. Okay, so that's the step. You after that you can see I want to follow the trajectory from this position along and to this. So that's your primary parse. Okay, so that's the algorithm to reconstruct trajectory using minimum spanning tree. So for this algorithm, the first step will be dimensionality reduction and put your cells in different clusters. Then you identify the center of the clusters and use those cluster centers as a vertices. Then you connect the center by by this. You calculate the distance between centers, then you reconstruct this minimum spanning tree, and then based on the tree, you found the major trajectories. And if you want to study another secondary trajectory, you can go from another parse. Okay, so this is one algorithm. So another algorithm is called nearest neighbor graph based. Okay, so for example, these two software tools using nearest neighbor graph. So once you have your cells and you compute the distance between cells, then you use nearest neighbor, okay, KNN approach to construct a KNN graph. So once you have this graph, you can follow the trajectory along this graph edges from one end move to another, and then you can reconstruct those from averaging of the short ways works. Okay. Well, as you can see, both these two software tools rely on the build of KNN graph. So I'm going to explain to you what KNN graph is. So this is for nearest neighbor graph, and it's a network then built based on the nearest neighbor of each data points, and you can predefine a parameter k, and so that it's become a KNN graph or KNN graph or KNN network. So if you decide I would like to search for nearest neighbor of 3 of each data point, then your KNN will be 3 nearest neighbor. Or you can define as 5, then your k equals 5. Then you have a slightly different version of your KNN graph can be built. So I'm going to use this online demonstration tool to show you the creation of a KNN network. Now we are on this page created by the Warframe demonstration project to show you how a nearest neighbor network can be constructed from a set of data points. So on this page, we have a predefined 80 random numbers, random samples. They're the right dots you can think about in our context. Their genes, their cells has been displayed in this two-dimensional tSNE space. And then we ask for to generate linkers between data points based on the nearest neighbor and how many nearest neighbor to join. And in this setting, we set k equals 1. So for each data point, we search for the nearest neighbor and make a link between them. For example, the nearest neighbor of this point is here, so we make a link from to here. The nearest neighbor of this link is this cell, this data point. So we make a link between them. And for this data point, the nearest neighbor is this one. So this link is mutual connected. Something like this is also mutual connected because of the nearest neighbor for each other. Yeah, they're nearest neighbor for each other. What I can do, make a adjustment is to increase number of k, to increase the connectivity, the linkers between data points. If I increase this to, see, number 2, and you see for each data point, it will search for the top 2 nearest neighbor and make a link. So you can see there are more linked nodes than the scenario when k equals 1. I can do further to link, make a k3. In this case, for each data point, you will search for the top 3 nearest neighbor and make a connection between them. And when you increase this K, you will see the increased level of connectivity among this network. Okay, so you can decide yourself based on application to increase or decrease your K in order to recover this structure of distribution of those data points. If you prefer to have a small K and then the connectivity level is relatively low, so you still can trace this trajectory. And if you increase this, you will have an increased level of connectivity, and then you still be able to search for the shortest path along this network. In any case, you only be able to link this to really far data points because you rely on the nearest neighbor algorithm to build this network. Okay, so let's go back to our slide. So in this example is a sample KNN graph, and in your left is K equals 3, and on your right is K equals 4. It's two different conditions. So when you have a small K, for example, in the data points inside this red oval, there are separated components. They're not fully linked because this is an independent subgroup of data points. When K equals 3, they are not joined to your with any other points. However, when you select K equals 4, because each data point will search for top 4 nearest neighbor, at the end you merge this isolated components into a larger network. So that's an influence of the K selection to the network establishment. Okay, in single-cell RNA-seq data, you can have this tSNE plot, and each point here is a cell, and then can apply KNN algorithm to establish such a network between those cells. So I didn't show the cell with these data points here, but only show this framework of this KNN network. So after you build this, you can start to use one of the starting point cell as a starting point to search for a path as long as possible, and from one edge of the network and to another. So you can go along with those linkers to build a trajectory and use this trajectory as your pseudo-time trajectory, and then it's possible to cover most of the cells and assign the values of a pseudo-time for the neighbor cells to this trajectory. Then you can use this information to look for gene with the expression associated with the increase or decrease of the pseudo-time. So that's an application of KNN in pseudo-time analysis and build this trajectory. You can find the source code for using MATLAB. You can reconstruct this network on live. So the link is here, and it's a GitHub repository. And there are other algorithms as well, and this so-called cluster networks, and also most recently their RNA velocity method is based on the splicing site and to infer the transcriptome change between cells in different states. Okay, so that's a principle. You can use KNN, you can use minimum span tree to look for the trajectory. So the next couple of slides, I'm going to mention some most commonly used tools for trajectory or pseudotime analysis. The earliest one is published in Nature Biotechnology in 2014. It's called Monaco. So that's version 1 of Monaco. It's pretty early. At that time, there are very few single-cell study. So in this first version of Monaco, they use ICA, which is another dimension reduction. We haven't mentioned about this methodology. You can have a high dimensional expression data sets and then reduce the dimension to show the relationship between cells. Then Monaco use minimum span tree to link those cluster centers. And then older cell in this pseudotime using MST information and then label different cell types and build this trajectory. Then for the downstream analysis. So that's an example of the application. And after build this minimum span tree, the black and the black edge, and then it assumes this is the beginning of the pseudotime, so you can follow this trajectory to do the analysis. And then you can have a branch out of it to do the separate analysis. Monaco then published version 2 a couple years later, and they changed the algorithm. And they use the so-called reversed graph embedding to do the reconstruction of complex single-cell trajectory. And claims that it produce more accurate and robust trajectories than its previous version. And they require no prior information as before. And so that's some description of the reverse graph embedding. It's based on so-called principal graph. Principal graph in this new technology. They use principal curve that pass through the middle of the data set that allow to have some branches. So they rely on this and to start to build this tree and then develop this new version of the Monaco. And then they apply this. Still, the first step will be dimensionality reduction. You can use different tools to do it. Then to build this using principal curve to found this trajectory, and then you assign these cells, move cells to the branches to the parts, and then you can further reduce the dimension and then do the analysis. So that's the figures from Monaco 2. Most recently, Monaco published version 3, and it's also focused on the trajectory analysis, and also emphasize it's a comprehensive package, can do clustering analysis and also differential expression analysis. But the clustering algorithm, they use UMAP to do the dimensional reduction, and then use the Louvain network community detection algorithm to cluster cells. However, in the field of single-cell RNA-seq analysis, people still regard monoclonal as the main dominant tool used for trajectory analysis. Okay, besides monoclonal, there are other tools that have been developed. So this is called the slingshot. Slingshot. It's based on the simultaneous principal curve. Okay. So you see both packages use principal curve, and this curve can help you to find this trajectory among cells. Then you assign each cells to the nearest branch, nearest parse. So I'm going to show you an example of using principal curve to do this fitting of the data points. So I have an example of a principal curve fitting. So I have run the first example. You will see they are let me quickly show you an example. I run the first example. So on the screen, you see make some adjustment. So on the screen, you see an example that you have data points, make this half moon shape, and then the green curve is learned from it using this principal curve algorithm. And then I'm going to show you the second example. And this is a cloud of data points. Not so clear about this structure, but you see this principal curve algorithm can find the middle of the data points and then to find out this trajectory. Okay, and the third example will have a different shape of the data points. And in this case, this particular algorithm can find multiple trajectories from the data points. And then the last example I'm going to show you will be two-dimensional. And then you see those data points form this coil shape, and also there's another separated parse in the middle of this coil. Okay, so that's what a principal curve algorithm can do. So if you have a single-cell RNA-seq data and you dimension after dimensionality reduction and put them into three-dimensional space, you can learn directly from this cloud of data where the structure, where the principal curve is. And you can use this principal curve as a pseudo-time trajectory. Okay, so that's a principle behind all these commonly used packages, monoclonal and slingshot. And the summary from the slingshot publication and compared to other packages like monoclonal 1, 2, and also there are other tools, and then tScan we'll mention, and they use different dimension reduction algorithm. Monocle 1 use ICA, this Monocle 2 use reverse graph embedding, and then tScan is another one, use PCA. Slingshot can use any input of results from a dimensionality reduction, and then it rely on clusters, and some of these are not rely on clusters. Graphs, some algorithms use MST, some use principal curve. Okay, so that's the features, and then how to calculate pseudotime, and there are different ways to do it, and whether allow the branching or not, and either the supervision, and whether you need a human intervention to tell where the starting point of the cell. And some of these require, some of these results require, some of them not. And the author of Slingshot also provide examples, say, okay, this method is more reliable, and this Slingshot create the more reliable outcomes compared to tScan and the Monocle, which this original pseudotime setup will hit this, and then you get the results not so well correlated. However, in Slingshot case like this, the correlation between the original pseudotime setting up with established is highly correlated. So this thing is very robust to the noise. And then they have some examples to show the Slingshot can do analysis for all the 3 trajectories, and the Monocle may only recover 2. As I mentioned, the tScan is another trajectory analysis package, and published in Nucleic Acid Research. They use PCA to do the dimension reduction, then use minimum spanning tree as an algorithm to create trajectory. It's unsupervised algorithm. Okay. This is another trajectory analysis package, and recently published. And that's the pipeline outline of their pipeline. Lisa is less commonly used, and they rely on K&N graph. Okay, you have the tSNE, and then dimensionality reduction, then use the K&N graph. Then they use a peak valley selection based on this ISO map, landmark ISO map, and then rely on minimum span tree applied to the cluster center to estimate pseudotime. Okay, so I have mentioned a couple of these commonly used or less commonly used trajectory analysis software tools. We really have a lot of them. So in this easy table, you see each row is one of the pseudotime or trajectory analysis method has been published. And this review paper actually collected 45 of different methods and compared their performance and to give you an overall evaluation. So in this table, they summarize the features of each of these methods, and they're using overall performance, accuracy, scalability, stability, and the usability as a measure. And they quantify all this performance and assign a value for each of these tools they evaluated. So that's only part of the table. The second half of the table is here. So you can see T-scan is here, and we mentioned wishbone, we mentioned waterfall, we mentioned, and the previous slide we have a slingshot, and the monocle should be somewhere here. Monocle ICA, monocle version 2, version 1, and version 2. So this is a comprehensive comparative study to evaluate all of them. In the then they have a detailed results of 4 main evaluation criteria. As I said, accuracy, scalability, stable, and usability. They put them in a more detailed table, and that's the second half of the table. At the end of this review paper, they provide a guideline, practical guideline for method users, and how are you going to decide which tool you are going to use. So they have some questions, whether what's the data type you have, what's the structure of the topology of your cells population, and then you can decide by asking those questions, and then they will point you to the best selection of the method. We can see slingshot, and the T-scan is here, and the monocle 2 is here. So that's a review paper for the methodologies. So you can use this as a practical guideline. There are another efforts in the put together all of these packages developed in R, all different trajectory analysis. They call trajectory inference method, and under the same structure, so you can run a lot of different methods in the same interface. It's called Dyniverse. Dyniverse is a collection of a lot of different tools and put them together. All these tools can be used to do the trajectory analysis. Okay, so trajectory analysis is once you construct, you can do a lot of downstream analysis. Remember, this is only intermediate steps. You establish the trajectory, and so you can align cells based on its trajectory, assign pseudo-time on it. Then you can do a lot of other things, and it depends on your questions, whether you want to study the topology, cell state, transition, trajectory. You want to compare the different ends of this trajectory, whether the gene have a clearly down or up regulation. And you can visualize the relationship between the changes. Anyway, trajectory analysis is advanced topic in the single-cell RNA-seq analysis, but still it's in the middle of the research, and you want to use the reconstruct the trajectory as a plug-in in the information, as add values to your data analysis. So that's all for today, and thank you for your attention. I will see you next time.